Well, thank you very much, and very unusual times of year to do this presentation by Zoom. But I do want to thank the organizers of the first electronic pharmaceutics meeting and the people at the Journal for the opportunity to speak today about some exciting work that we've been doing in our lab and with iCo-Therapeutics over the last number of years. So the title of my project is the development of a novel oral Amphotericin B formulation to treat systemic fungal and parasitic infections. My name is Kishore Wasan. I'm a professor in the Faculty of Medicine and Distinguished University Scholar at the University of British Columbia, but I also play a role as the Director of Research at iCo-Therapeutics, where we license our oral Amphotericin B formulation for clinical development. So let's jump right into it. So Amphotericin B is a Pauline macrolide antibiotic used in the treatment of blood-borne fungal infections and parasitic infections like Lishmaniasis. However, the drug is poorly water soluble. And in fact, there's a number of issues with Amphotericin B in that it's tropically not stable. You have to give it intravenously. It's not cost effective. And it has renal toxicity and other systemic toxicities. So the thinking was, could you develop an oral formulation of AMP-B that's bioavailable enough, cost-effective, safe, and that could be accessible at pennies a cost that you could give to people who have systemic fungal infections and terrible parasitic infections that you find in the developing world. Now, it's really important to know the mechanism of action of Amphotericin B because this is going to be important that helps us frame how AMP-B, we reformulated it and used it in drug delivery. So AMP-B is what I call a membrane disruptor. It binds to the sterol component of either fungal cells, parasitic cells, or mammalian cells. And it's the monomeric form of Amphotericin B that it comes and it binds to that sterol component and lyses the cell. And that's how it causes its toxicity. As I mentioned before, many of the infections that Amphotericin B treat are really in patients that are immune compromised. So there are patients that have got either post-transplantation, patients that are uncontrolled diabetics, cancer patients, patients with HIV/AIDS. We've even seen recently, unfortunately, patients that have had COVID. So patients that are immune compromised, that are susceptible to these systemic fungal infections and parasitic infections can be treated with Amphotericin B. And here's a whole list of different infections where AMP-B would be the drug of choice. And the beautiful thing about Amphotericin B is that there's very little drug resistance, that the drug is actually quite effective, but the big problem is around its delivery. So here's a number of different disease states that it can be used for. There's azole-resistant candidiasis infections and VVC in women, recurrent VVC, and it goes on with some histoplasmosis and several others, and I won't go through the whole list. You can do this yourself. But another one that's very important is visceral Lishmaniasis, and Lishmaniasis is a devastating parasitic infection. In fact, we're looking at maybe over 250 million people worldwide that have been infected with leash in one form or another. And the problem with Lishmaniasis is it happens in developing world countries or in countries where there are poor sanitation conditions. And there is a sandfly that comes along in these poor sanitation conditions. It stings you, releases the parasite, which goes primarily into the lung or to the spleen, and will cause the growth of proliferation of this virus or this parasite in these organs and can lead to death. And so the current drug treatments for Lishmaniasis are things like paromomycin, which is an aminoglycoside, but it's an IM rejection. Miltefosine, which is the only oral drug for the treatment of leash. However, it's got teratogenicity. On top of that, it is tropically not stable. And in fact, it breaks down quite a bit and it's hard to get to patients. So the great opportunity was, could we develop an oral formulation that could overcome some interesting barriers? And some of the most things that are holding back parenteral AMP-B that an oral product could maybe deal with is loss of income due to hospitalization from IV therapy. So intravenous Amphotericin B, the lipid-based Amphotericin B products, and I worked on Abelset as a graduate student, so I'm very familiar with these. are fantastic drugs. But the problem is, in the developing world, where people do not have access to hospitalization and place where you need to have IV infusion, it's hard to get these drugs there. So accessibility is a big problem. Second is the high cost of administration, though now there are generic forms that make it a little bit cheaper, but it's still costly. It's not only the drug treatment, but having trained personnel to give you the infusion, as well as having an IV bag, a trained facility. So there's all these additional costs with any type of intravenous or parenteral administration. There's the risk of infusion-related side effects with the drug, which you can get around with an oral formulation. There's the risk of systemic toxicities. We found that if you give an IV infusion of Amphotericin B, patient gets shivers and chills from giving that infusion too quickly. We can get around that by giving an oral form. And finally, there's no heat, it's not heat stable. So you have to have a cold train. You have to have it in refrigeration. And in the developing world, we just don't have those facilities. So if you could have a tropically stable form of Amphotericin B, that would make a huge difference. So one of the things we've overcome with our new formulation is easy to administer at home. You don't have to have the trained personnel because you can take it yourself, and drug adherence, decreased cost of administration, the lack of infusions related to side effects, as I mentioned, the lack of kidney, liver, and GI toxicity, and the increase of accessibility, as well as thermal stability at tropical temperatures. So the first set of studies that we did was visceral Lishmaniasis studies, as well as antifungal animal studies. And so we developed a formulation, which is a really is a mono and diglyceride reverse micelle formulation that we have developed. And one of the first studies that we did was actually funded by the Bill and Melinda Gates Foundation. And they had a validated VL, visual Lishmaniasis model in mice that they use as a screen to test many drugs. And so we worked with the Gates Foundation through Ohio State University that had this model in place to test our formulation. We got amazing results. So what we have here on the y-axis is the LDU units, the leash parasite units that is found in the liver. And on the x-axis are different treatment groups. We have the untreated group. We have the vehicle control, which is the formulation but without Amphotericin B. We have miltefosine, which is the positive control, which we gave at 3 mg/kg PO QD times 5 days. And at this time, that was the kind of dose that was actually administered in the clinic and on the ground. Then we had ambisome, which is the liposomal formulation of AMP-B, the parenteral formulation as a positive control. And you could see that it was very effective at 2 mg/kg. But what was exciting for us is that our formulation at 10 mg/kg and at 20 mg/kg pretty much eradicated and cleared 100% of the parasites. 60% of the animals had 100% clearance of the parasite. And so even though we have only an oral formulation, we were almost as effective as ambisome, which was really exciting. And the fact that now we can have an oral formulation that could overcome the limitations of the parenteral formulation. In this next slide, we're showing you the same thing, but we showed you as a dose response. And so again, the LDU is on the y-axis. This is the amount of parasite that's loaded up into the liver. Here we have the vehicle control. And then at 2.5, 5, and 10 mg/kg, BID for 5 days. And what we found is that we had an ED50 of less than 2.5 milligrams per kilogram with our formulation. And in fact, we share you one of the immunostains. The blue represents the amastigotes of the parasite. It also is a non-specific stain for the nuclei. So that's why you have the nucleus in both control and treatment group. But look at all these little amastigotes of VL in the untreated group. And look at what happens with our formulation. Maybe 1 or 2, almost completely clear. So this was really exciting results. And in fact, this paper was published in the Journal of Infectious Disease a number of years ago. But what was really important is this was done in an independent lab at a third-party lab validated by the Gates Foundation and their labs, independent of the work that was done in the Wasan lab. The next thing we did, or other studies that we did, was really look at the efficacy of our oral formulation and ambisome in our animal models of antifungal. But what was also really important is to look at our formulation prior to it being exposed to tropical temperatures and then after it had been exposed to tropical temperatures, and see if we can maintain the efficacy of the formulation. We made the claim that we had a tropically stable form of the formulation to use AMP-B to treat these diseases. Does it actually work? And the exciting results was it does. So what we have here is the colony forming units on the y-axis. So what we did is we infect our animals with Candida albicans for a period of time, let it seed into the tissues, and in this case, most of it actually seeds in the kidney, and that's where most of the infection occurs. And then we measure the colony forming units in each of these different treatment groups. So the control group is up here. You can see that we have a pretty high control group. The AmBisome, the positive control with an IV infusion at 5 mg per kg, reduced that fungal load by 97%. But what was really exciting is our formulation at 5, 10, at 20, our tropical formulation, iCo-019, had a significant reduction in the fungal load. But more importantly, it didn't change regardless if you gave it fresh or after you incubated it at 43 degrees C for 60 days. In other words, it was tropically stable. It still performed very well. And again, this is another study that we did showing significant antifungal activity in the rat model. So this is another model now, where again, we infected the animals with Candida albicans, let it seed in the kidney. And what's the nice thing about these animal models is that they do actually mimic what you see in patients. That Candida albicans quite a bit ends up being seeded in tissues like the kidney. And so this is really a good reflection of what would happen in the clinic. So we have fungal infection in the kidney here, the CFUs, here's the control, and then our formulation once again has a really great reduction in the colony forming units when we gave our formulation. And the Amphotericin B lipid complex was a positive control. What was also really exciting, AMP-B, as you know, is a nephrotoxic drug, but in our formulation, it was able to reduce the fungal load, but it did not affect the renal function of the kidney. In other words, there was no change in plasma creatinine levels, which is an indirect measure of renal function. Again, we show you in another model of Candida albicans the significant reduction in activity as a function of dose. And what was really important is the biodistribution of Amphotericin B in mice following multiple dose treatment with our oral formulation. And what people don't appreciate is AMP-B has some very unusual pharmacokinetics. It is not about plasma levels or Cmax levels. So many drugs, you correlate Cmax or plasma levels to efficacy. Amphotericin B, really it's about tissue distribution and being able to have the drug go into the tissues and to have a sustained depot effect. So in fact, the clearance of drug from the bloodstream into the tissues is faster than the clearance of the drug from the tissues back out. And so if you can build up drug into the tissues over a period of time without having toxicity to those tissues, it can elicit its pharmacologic effect. So what I'm showing you here is that when we administer our formulation for 20 mg per kg twice daily for 5 days, 10 mgs or 5 mgs or 2.5 mgs, we're measuring the amount of drug in nanograms per gram tissue concentrations over that period of time. And what you see is a buildup in the amount of drug in those tissues as a function of dose. And since the MIC is in that 20 nanogram level, we are way above the minimal inhibitory concentration required to have pharmacologic effect. And so that's why the oral formulation is effective, is that we're able to build up the drug into the tissues as a function of time. And here's IV Fungazone as a positive control. And Fungazone, of course, since it's directly IV, is going to have a huge amount of drugs in the tissues, but you don't need that much to have pharmacologic effect, you know? And in fact, you get toxicity with the IV formulation of Fungazone. Not only the renal toxicity, but the systemic toxicities because of the IV administration. So this is really, really important to understand about the unusual pharmacokinetics and biodistribution properties of Amphotericin B. Now, what's really exciting is the following. And you might ask yourself, you know, this data doesn't make a lot of sense in that when we gave our oral formulation to animals, Here's an animal untreated where the Candida albicans basically engulfs the kidney. And yet, after we treated it with our formulation, we were able to remove the fungal infection, but we were able to spare the kidney. So the kidney was not toxic. Now, you might ask the question, but that doesn't seem to make sense. Amphotericin B is a nephrotoxic drug. So if you target the kidney with AMP-B to kill the fungal infection, wouldn't you also damage the kidney? What's going on here? Why is this so special? And in fact, here we're showing you some histopathology where, again, our oral formulation, there's not much toxicity. There's nothing in the liver. There's nothing in the jejunum giving it once you give it orally. Yet the IV, you can see the necrosis here. Here you can see the liver necrosis from the IV of fungizone. And of course, there won't be any toxicity jejunum because we're giving it as IV. But this asks the very important question: what is happening here with amphotericin B? Why is it able to, be able to kill fungal cells and destroy them, but spare kidney cells and not hurt the tissue? And the reason is because fungal cells release what we call phospholipases. And what these phospholipases do is they cleave the amphotericin B from that drug delivery lipid package in the monomeric form. Remember, I showed you that it's the monomeric form of amphotericin B that binds to the sterol component of the cell membrane and lyses the cell. So it has to be in that monomeric form to be effective. All right, when it's not in that monomeric form, it will not bind to a fungal cell or a mammalian cell, and it will not elicit damage to those cells. So what I'm showing you here is actually some interesting data from kidney cells. And what we did is kidney cells, if you don't have any phospholipase, it has only a certain percentage of damage to the cell. This is represented in the blue. But when we add phospholipase to kidney cells that normally don't have phospholipase, look how the toxicity increases. And so this data suggests that, in fact, it's this phospholipase mechanism that's involved in breaking down amphotericin B into the monomeric form. So what happens is that the formulation, by the way, liposomal AMP-B works in a similar way. The formulation comes with the drug package to a fungal cell. The fungal cell releases these phospholipases, which breaks down the AMP-B into the monomeric form, which inserts itself into the membrane and destroys the fungal cell. However, kidney cells do not have phospholipases. They don't secrete them as a protective mechanism. So what happens is when the formulation comes to the vicinity of the cell membrane of a kidney cell, it can't break down the AMP-B or release the AMP-B into the monomeric form. And therefore, the AMP-B cannot bind to the sterol component of the kidney cell and lyse the cell. So therefore, you're able to systematically save the kidneys from being damaged and the kidney cells from being damaged, but you could kill the fungal cell. And this unusual formulation approach and what we see in nature really helps us come up with a really great formulation to treat these diseases. The other question is, how do we get the sustained absorption of amphotericin B and this tissue distribution? How do we get this distribution into the bloodstream that then is at a high enough concentration that it will go then into the tissues and build up into the tissues as a function of time? And one of the mechanisms that we've used is lymphatic transport. And there's been some amazing work by the Monash group at Monash University down in Australia, Chris Porter and his team down there, who really are the world leaders on lymphatic transport. But we've done some work as well. So what is lymphatic transport? Lymphatic transport is the following, and it's really like when you have a high-fat meal or a high triglyceride meal. It does the same thing. What happens is that lipids and drug will come into your GI tract. Instead of being absorbed from the portal vein in the GI tract, from the GI tract into the portal vein and taken into the liver and then passed out from first-pass metabolism, what actually happens is amphotericin B formulated in these lipids can be taken up through what we call the mesenteric lymph duct. So chylomicron-like particles will bypass through the GI, the GI membrane, and will come into the lymphatics, into the mesenteric lymph, bypass first pass metabolism, the portal vein, and the liver, and be taken up into the bloodstream. What's also very interesting is the mesenteric lymph flow is much slower than the flow rate of the bloodstream. So what happens is when you have an oral formulation that's chylomicron-like in nature and has drug packed into it, you can have drug coming through the mesenteric lymph and be sustained in its absorption over a function of time as chylomicron-like particles into the bloodstream. And then these chylomicron particles can actually be taken up into the tissues. So this is really one of the mechanisms we've used to improve the absorption and the tissue distribution and the tissue depot effect of oral amphotericin B. Now, what I'm showing you here is the model that we've used. It's a rat model, and we have the ability to co-cannulate the mesenteric lymph duct as well as the thoracic lymph duct, and you can see that here. If I go back one slide, here's the mesenteric lymph duct, the cannulation for the mesenteric lymph duct. And what we can do is we can follow the fate of the lipid and the drug from in the lymphatics, in this lymphatic system, as well as how much is coming out on the other end in the bloodstream. And this tells you that, in fact, we are targeting the lymphatics. This actually shows you a table that shows that the transport of amphotericin B in rats following an oral gavage of our formulation, which is really a combination of PCL, which is a mono and diglyceride mixture, compared to suspension control. So what we're showing you here is the following. This is following an oral gavage of 5 mg per kg of amphotericin B to patients. What we're showing you here is the amount milligrams of AMP that's transported lymphatically by time interval and the total lymphatic output. So the suspension formulation has a little bit going into the lymphatics in the first 4 hours, in the 4 to 6 hours, but by 6 to 8 hours, it really trickles down to almost none. And here's the flow that you get in that 8-hour period of time. However, look at our formulation. You get more than double in the first 4 hours. It continues to be more than double between 4 to 6 hours. And look what happens from 6 to 8 hours. It's almost 12 times more drug being transported through the lymph using our formulation. And so what you're doing is, this data suggests that we are targeting drug through the lymphatics, but it's sustained over a period of time. And that gives you the pharmacokinetic profile as well as the ability to get into the organs and have a depot effect. So in summary, what do we have here? We have potential mechanisms for a lack of renal toxicity and enhanced drug absorption. We have a drug delivery package that makes AMP primarily available for fungal cells through this fungal phospholipase mechanism. We have lymphatic transport, which increase bile salt secretion, and we can target the drug through the lymphatic transport. It bypasses first pass metabolism in the liver. We get more drug in the systemic circulation, but over a sustained period of time. So, and we'll show you that in a few minutes with the PK data, and that it will go into the tissues. And it's really this tissue distribution that's really important. We've enhanced the solubility of AMP in this formulation. AMP is an acid labile drug, so that if you just gave it orally as is, it would break down. The OH groups on the backbone of amphotericin B would be cleaved, and it'd be like sand, and it would never be absorbed. But we've been able to enhance the drug solubility by putting it in our lipid package. And we've been able to enhance the stability at the gastric pH, as I just mentioned. Therefore, it is the active compound, not a broken down compound, that's available to be absorbed and have its pharmacologic effect. And this is really a summary of the many animal studies that we've done over the years with different formulations. And you can see we've done a mouse study in VL, we've done a rat study with Aspergillus, we've done several candidiasis studies, we've done biodistribution. But I think the take-home message from all these studies is the following: is that number one, we have a biodistribution available drug that can have a significant pharmacologic effect without the IV toxicities associated with an IV form or the kidney toxicities associated with the drug. And the fact is that it's a tropically stable, cheap, and accessible kind of formulation, which is a game changer in the field. Over the years, we needed to reformulate it. Now that we've got this great animal data, we needed to go ahead and reformulate it, not only for intellectual property purposes, but ultimately we needed a formulation that we could give to man. Up to this point, we had a liquid formulation, which is fine, but liquid formulations have pros and cons, and we really wanted to have a stable capsule formulation that we could give to patients. And so what we're summarizing for you here is that we did a number of studies a number of years ago comparing our lipid formulations and liquid formulations, and then we settled on an oral formulation, which is called an oral capsule formulation, which is called iCo-019. So unlike iCo-10 and iCo-9, which were liquid preparations, this is a capsule formulation. And here we compared the Cmax, Tmax, AUC, and MRT last of these different formulations. And the reason why we went with 019 is because it was easy to manufacture, it was easy to scale up, it was relatively cheap, but it had similar pharmacokinetics and toxicity profile that the liquid formulations had in beagles. And so we did, of course, a beagle study, a GLP tox beagle study, which we presented at the AAPS meeting in New Orleans about a month ago. But also, this paper has just been published in Antimicrobial Agents of Chemotherapy back a couple of months ago, if you want to get more details. And what we did is we basically looked at the pharmacokinetics, tissue distribution, and toxicology profiles in a GLP condition of our liquid formulations versus our capsule formulation. And what we saw was very similar plasma levels, no similar lack of toxicity, not only renal toxicity, kidney toxicity, liver toxicity, but we also looked at red blood cells. We looked at coagulation products. It was a very detailed set of studies, toxicology studies. We did histopathology. And we saw that we could get the tissue distributions that we were looking for, and we did not have the toxicity associated with it. So this is one of the beagle studies and the ranges that we did and the tissue distributions. There was both a non-GLP toxicology component and a GLP toxicology component. And the other thing we did is when we moved this formulation into a capsule formulation, it was incredibly important that we did stability studies to maintain that tropical stability that we showed with the oral liquid formulations in the past. And the great news is that we have 24 months of data with the formulations very stable at 40 degrees C. And that's really important. So again, we wouldn't need to have a cold train when we gave this in the developing world. And what we're showing you here is some of the blood levels following oral absorption of our oral administration of drug to beagles. What is really important is the sustained AUC, okay? In that, what is important here is that our AUC is quite sustained over a period of time. And that means that we have more drug available in the systemic circulation that can distribute into the tissues and build up that tissue depot effect. And that was actually really important. So we did biodistribution studies and we didn't see any observable toxicity. We had comparable PK. We were able to do some pretty high doses to show that there was no issue there. We did fed fasted and really didn't see much difference in fed fasted. And we did 7-day and 14-day, 7-day non-GLP and 14-day GLP studies. And we're able to get high doses without the toxicity, but that sustained profile. And that led us now to our phase one clinical trial. And we had really two clinical trials. We had a phase 1A clinical trial and a phase 1B clinical trial. So in the phase 1A clinical trial, this was a placebo-controlled single-dose ascending study to assess safety, tolerability, and bioavailability of oral AMP-B in healthy male and non-pregnant female subjects between 18 and 55. And our primary goals were to evaluate the safety and tolerability of multiple dose levels. So we did from 100 to 800, and these were single dose studies. And we assessed the PK and the bioavailability after these single-day studies. And we had, in each cohort, we had 8 patients. 6 were randomized to receive the formulation, either at 100, 200, 400, and 800. And we had 2 randomized placebo controls. And all subjects were followed for 7 days after the dosing as well. And the results were really, really positive in the following way. We really met all the criteria. The study met its primary endpoint of safety and tolerability. We did not see any renal function, renal damage, or liver damage, or GI side effects at all. We didn't see any adverse events over the single dose period of time. And the other thing was the pharmacokinetics and the bioavailability oral AMP after a single dose administration. And again, it was this AUC that's really important. And what I'm showing you here in this table is that PK data. So here I've got the dose, the Tmax in the median range, the Cmax, the AUC to T-last, and the T1 half-life. And what's really important to notice is that our Tmax is about is between 6 to 7 hours. If you remember the lymphatics, in the lymphatic animal data, you saw that we got the highest amount of drug going through the lymphatics at the 8-hour mark. So we're getting that sustained Tmax correlating to what you see in the lymphatics. All right, the Cmax is pretty steady, but like I said before, it isn't about Cmax in terms of activity. It's about AUC, because what's happening is, as the drugs are coming in from the lymphatics, which is at a slower flow rate, and getting the bloodstream, it's leaving the bloodstream into the tissues a lot faster. So therefore, your Cmax is remaining pretty steady. But what's more important is the sustained AUC and the sustained T half-life that you're getting with the formulation. So as patients are taking this in multiple doses, as we'll show you in the next study, they're going to get a sustained exposure of Amphotericin B, but without the toxicity associated with it. And so that was really an exciting result. The second was to do a clinical phase B trial. And in this 1B trial, we did multiple doses. And the primary purpose of this trial was to look at to do a single-centered, double-blind, randomized study to evaluate the safety, tolerability, and pharmacokinetics of 100 and 400 milligrams, or placebo administered for 10 days in patients. And so what happened is that we evaluated safety and tolerability of repeated administrations of either 100 milligrams or 400 milligram doses of oral AMP for 10 consecutive days in healthy patients. And we looked at the PK and we looked at the toxicity profile as well. And again, we hit our primary endpoints in this multiple dose study. What was again very, very important is that we did not see any adverse events or drug-related toxicities. There was both not and repeated on drug doses was very well tolerated. There wasn't the GI toxicity, there wasn't the kidney toxicity, there wasn't the liver toxicity. But what's really important is again the sustained AUC as a function of time and dose. And this work was recently we said submitted, but since then was also presented at the American Association of Pharmaceutical Science meeting, and the 1A study was also published in Antimicrobial Chemotherapy a few months ago. And this is what one of the plots look like. So these are five patients, but look what's really important. Not only what we get to the Cmax, but look at the sustained over 24-hour period of time. And that's at the 100 milligram dose. And the 400 milligram dose showed a similar thing. In other words, we're able to get sustained exposure of the drug to the patients over a period of time to deal with the fungal infections or the parasitic infections, but without the toxicity associated with the drug. And again, this is what happens when you go for a little bit longer period of time, over 10 days. So the first one was after the first day of dosing, and this is after 10 days of dosing. And there's some more variability here, but the bottom line is in most patients, that sustained blood level and sustained AUC. And here I show it to you in the table form. And what's important here is that at the 100 milligram dose to patients, look how we have a doubling of the AUC over a 10-day period. And look at the Tmax being, or the T1 half being 24 hours and even greater. So this was an important finding and is consistent with the pharmacokinetics that we saw in our animal models. So what do we have here, ladies and gentlemen? We have a formulation that's designed specifically to overcome one, the limited absorption of AMP from the GI tract. We use both the way it was formulated and protected from breakdown in the GI tract and solubility, and lymphatic transport to increase that sustained absorption and biodistribution of the drug. And two, we have a formulation that gets over the toxicologic issues for fungal infections. So AMP is not amphoterrible in terms of treating dealing with kidney toxicity because we've packaged it in some way. Plus, we get around any of the IV infusion related issues of giving the drug as a parenteral formulation. We have tropical stability, as we mentioned, 24 months out to 40 degrees C, and it's a proprietary blend of mono and diglycerides, which is FDA GRAS approved, and that's really, really important because the excipients themselves are very, very safe. They have FDA approval, and so the toxicity profile is acceptable to the FDA. And really, we have low cost of goods compared to injectable. It's easy formulation to scale up. It's relatively few steps in the formulation, and the excipients are affordable. So it's pennies a day to treat patients, particularly in the developing world, and it's accessible. They can take it by mouth, so the drug adherence goes up. The other big advantages of the oral formulation is this would be the first fungicidal agent on the market. To date, there is only fungal static agents. That means that they will prevent any further growth of a candidiasis infection and so on, but they can't kill existing fungal infections. Our formulation can. And the big thing about oral AMP, right now fluconazole is the drug of choice for VVC in women, but it's only got about 50 to 60% effectiveness, and there's a greater than 9 to 15% of patients, women, who get reoccurrence. We think our oral formulation will be able to be an advantageous therapy over fluconazole. And we're looking now to do a phase 2 clinical trial in VVC where we're going to do the head-to-head against fluconazole. We've had orphan drug designation from the FDA for a number of years now, particularly for VL. We have a number of patents issued, though we've now got some new enabling patents that have just been issued, that will be issued, and that will get us to the 2030 range. We have, as I said, completed our clinical trials, 1A and 1B, that I shared the data with you, and the papers have been published. And it's easy to store and easy to administer. And here is the product profile for our formulation. I won't go through this all again because I've already described it. But I think the main thing that's important is that it's an oral capsule that is tropically stable, that's easy to give, and has pharmacologic effect. And we have a IP exclusivity for another good 17, 18 years. And I think that this can be a product that can be used for multiple things. I mean, it could be a product to treat patients secondary to systemic fungal infections. It could be there to treat patients who have visceral Lishmaniasis. It could be there to treat patients as a first-line therapy for things like vaginal candidiasis in women. So the flexibility of the oral AMP product is great. I mean, it's safe, it has broad spectrum of activity, but it has uses in multiple ways. And that, I think, gives physicians extra tools in their toolbox to treat with these diseases. So I come back to this slide that we had in the beginning, which is what was the advantage of having an oral AMP product? Why over a parenteral AMP? Again, it's easy to administer at home and to store at home. Okay, you don't have to have it as an IV. This will increase patient compliance. You can take the medication to people. They don't have to come into the hospitals. And people that live in rural communities, particularly in the developing world, where they may have to walk hundreds of kilometers to get to a hospital or to a clinic, we can take the medication to them. Decreased cost of administration. It's not only the cost of the tablets itself, but all the other aspects to doing a parenteral formulation. You don't have to worry about the IV bag or the needles or the trained personnel or a sterile condition for an IV. We can give this as an oral formulation. You have a lack of infusion-related effects. This is given orally. Again, IV infusion of AMP has to be in a monitored hospital environment, and you have to monitor the infusions because you can get a risk of infusion-related side effects. We have a lack of kidney, liver, and GI toxicity. We have increased accessibility, all right, and drug adherence. And finally, we have a formulation that's tropically stable. And so these are just the references at the end. But I think at the very end, we've got something that we're very excited about. And we're looking forward to doing phase 2 clinical trials in the upcoming year. And I want to thank you very, very much for this, for giving the opportunity to do this presentation, and to the pharmaceutics group for letting me do this recorded presentation. And there is an abstract as well as a copy of these slides on the website. And if you would like to, you're more than welcome to access it, as well as send me any questions. So with that, thank you very, very much, and you all have a wonderful day.